main viruses that we're going to use is the HIV virus as a model. So we want to look at the HIV as a model virus. Not meaning that it's like the best virus or the only virus, but focusing on the fact that it has some really unique characteristics about it, that if you study HIV, you understand a lot about what a virus is in general. So HIV was first diagnosed or characterized in 1981. It does not mean that it didn't exist before then. It just meant that this was when the first human cases were recorded. And so what this tells us is the idea that this virus probably existed before 1981, but that it actually evolved or changed over time so that it could infect humans. So the idea of evolution, and we do want to go back to what does evolution mean? Evolution means a genetic change over generations. So yes, a virus can evolve. If it can change genetically, it's going to evolve. And so the idea that at some point this virus was probably infecting another species, another group of organisms, and there was some sort of random mutation that made it now able to attach to human cells. And so that's a really important characteristic of HIV, is this idea of specificity, but also the idea of evolution. And we see this evolution really continuing as you look at the drugs that treat HIV. The idea that if we're kind of selecting, if we're putting a selection force out there, we're going to almost encourage natural selection and evolution, because we know that HIV is going to have a high rate of mutations, because it is a retrovirus, meaning it's an RNA virus, and it's going to inject its RNA. It's really hard to see in this picture, but hopefully you drew it. It does actually also inject reverse transcriptase, ending in ACE. We know it's an enzyme. So this is a unique virus in that it injects its nucleic acid, but it also injects this enzyme. Now, it didn't make this enzyme. It tricked your cells into making this enzyme. But this enzyme is necessary in order to go from RNA backwards to DNA. So it is a retrovirus. We have a high rate of mutation. It's also a lysogenic virus, meaning we can have a period of what's called latency or dormancy where it hides. This is why you want to be really careful when you make friends, because you don't know who has this virus in them. You can't see it. There are people who never become AIDS. They're HIV positive, but they're never showing any actual symptoms because their virus is still in dormancy. And so this idea that we can actually test for the virus on people who aren't even showing symptoms. This is hugely important for the idea of transmission. Why is HIV so prevalent? People don't know they have it. There have been some very rare cases of people who know they have it purposefully passing it on. They have actually charged people with a crime because of that. But most people don't know they have it, but they're making friends and passing on this virus. And it also becomes really important for women to get HIV tested when they're pregnant, because if you give them, we'll go ahead and talk about this cocktail of drugs. A lot of this is because of the high rate of mutations. If you looked at the research I asked you to do, there's a whole huge class of antiviral drugs. Drugs that stop it from binding. Drugs that stop it from merging. Drugs that stop it from replicating its nucleic acids. Drugs that stop it from replicating these proteins. Drugs that stop it from budding out. There's this whole host of drugs, of course, because each virus, every time it reproduces, is mutating. No one drug is 100% effective against this. However, if you put a woman on the HIV cocktail of drugs, she's much less likely to pass it on to the baby when the baby is born. The baby doesn't get it by being in the placenta. The baby gets it because when baby is born, baby's exposed to mom's fluids, period. There was a really cool recent case of them actually being able to cure a baby of HIV. The baby was born with it. They weren't able to prevent the transmission because mom didn't know she was pregnant. They put the kid on the cocktail. They saw remission or almost all latency. And the last test, they were actually able to show no trace of the HIV virus. Does that mean it's completely gone? No. But they've labeled this child as functionally cured. So high rate of evolution because of this high rate of mutation. The idea that it's lysogenic and that it can hide. But the other idea is that it attacks your own immune system. So the cells that it is specific for are actually very important cells for immune function. So people who are HIV positive, who develop acquired immunodeficiency syndrome, who acquire AIDS, who start showing symptoms because their T cells are being damaged, these people don't die from HIV or AIDS. They die from opportunistic infections. And so that is a really interesting side effect of HIV, is that you don't die from being HIV positive. You die because you acquired these diseases that your body should normally be able to fight off if you had a functioning immune system. So they do actually want you to know that there are certain drugs that can block any of these pathways. One of the most famous ones is called AZT, and this blocks reverse transcriptase. And that was kind of one of the big first ones that they really discovered. We do want to talk just a little bit more about some other examples of viruses. So we're focusing on what other viruses threaten humans, but you do want to keep in mind, viruses can infect any cell. If they can bind, if they can inject, they've infected the cell. Some viruses have been contained or eradicated. So the main one that we see being eradicated is really smallpox. Of course, there are samples of smallpox left. They're in storage in the Center for Disease Control in Atlanta and in a facility in Russia. And of course, there is the worry of bioweapons. The main way that these have been done are vaccines. Keep in mind that there are really no 100% antiviral drugs. Now, a vaccine just doesn't cure you of it, but it means that you can't get sick from it, so there are no carriers of it either. Keep in mind, in order for a virus to really be effective, it has to spread not just from cell to cell, but also from host to host. So the idea of how do you get from host to host, or what's called host transmission. So one of the big things that epidemiologists, people who study the transmission of disease, one of the big things they do is try to decrease transmission from host to host. One of the ways they do that is actually through isolation. And that's one of the ways that they had to do that for smallpox. So they had to completely isolate people who were infected. One that they're working on right now is the polio virus. It's almost completely been eradicated. It's definitely been contained, but keep in mind, variations, mutations can occur. The influenza, this one is unique because of the high rate of mutations. We see this idea in the yearly vaccine. Influenza is an interesting virus. Here, of course, we see the capsid. This capsid is going to bind to lower respiratory cells and cause a lower respiratory infection versus the rhinovirus, which causes an upper respiratory infection. So just the idea that we have a variety of viruses that bind to a variety of cells, this idea of mutations keeps coming up. It's really important. Keep in mind that a mutation in the DNA or in the nucleic acid is sometimes going to transmit, this is why we'll put a question mark, to a change in the protein. So we don't want to say all mutations have an effect. We do want to keep in mind point mutations, frame shift, silent, missense, nonsense, all the different types of mutations. Ebola and SARS are important examples of emerging viruses. We kind of already talked about this a second ago when we spoke about species specificity. So there's a phrase for you, species specificity. And the idea that you can jump from species to species if you manage to match the receptors. Another one that is really, really common in the news is the human papillomavirus, abbreviated HPV. And the unique thing about the human papillomavirus is that it can actually, once it infects your cells, it can damage your DNA. So they found this unique virus that when it infects the cells of the women's cervix. Now men are carriers of HPV. They don't have a cervix. They carry the virus in their bodily fluids, but because they don't have a cervix, none of their cells are actually infected. If you're a woman and you have a cervix, and you get exposed to HPV, that virus can invade your cells and actually not just get the cells damaged by the virus, which we can kind of see here, but also we see it being integrated into our cells. So this tells us that it's a lysogenic virus. So we do kind of want to circle this idea of integrated, meaning that it's lysogenic. So it is going to incorporate itself into the host DNA, and this can actually cause cervical cancer. If we know what HPV looks like, we can create a vaccine. So this is the vaccine that's been created, and they're encouraging not just young women, but also young men to get it. As a child, you get dozens of vaccines that you don't even know about, and your parents don't even know about, and you're probably never even going to be exposed to it anymore.
Good afternoon, Butler students and staff. I hope you had a great Tuesday. I've got several announcements this afternoon, so I hope you're listening carefully. A reminder, first of all, for all those students who have after-school detention today that was assigned by an administrator, it will be in room 501 at 225. 501. Bus changes for today. Bus 761 is running for bus 1345. Bus 761 is now running for 1345. Bus 1648 is here for 1340. Bus 1648 is here for bus 1340. Congratulations to the boys golf team on their first victory of the season over Rocky River on Monday by 49 strokes. Top performers were Blake Estridge and Chase Britt. Good luck.